Welcome to our podcast on blood. As we begin, we're going to look at peripheral blood smears, and the most abundant of our leukocytes that we're going to see in a peripheral blood smear will be neutrophils. In this field, we can see a number of erythrocytes. We can also see these smaller structures that are the platelets. Here we can see the light-colored center of our biconcave erythrocytes, and you won't see this on every single one. But here we see the multi-lobes of our polymorphonuclear leukocytes, or PMNs, polymorphonuclear neutrophils, and we can see that we have fairly small specific granules inside and really a neutralish color. Sometimes they're going to look fairly similar to the color that we see of erythrocytes. So these are our neutrophils. Now once again, we can see a neutrophil in the upper left here. Lobe nucleus, small granules, fairly, and these look a little bit redder, but with every stain it's going to look a little different. Here this looks a little bit more reddish, but again, the size of the granules still are relatively the same. So these are both going to be our neutrophils. But then as we look in the lower right, we can see larger granules, more reddish in color. Certainly here we have a bilobe nucleus. This is a little trickier. I still think these are bilobed, even though it looks like they're three. But we can still see these much larger, more pink to reddish granules. These are going to be two of our peripheral blood eosinophils. Now in this slide, a little bit higher mag than we've seen in the others, we can see our erythrocytes. And remember, these are approximately 8 microns in diameter. So here we have a neutrophil. We can see the multiple lobes of our neutrophil, the small granules. On the far side, we can see a much larger cell. Really no granules to speak of, but since it's so much larger, this is going to be the largest of our cells. In peripheral blood and circulation, this is a monocyte. And remember, when this leaves the circulation, we call it a macrophage. Well, now for comparison purposes, we can compare the size of this cell in the center to our neutrophil. We can see it's a little bit smaller, possibly still bigger than a red blood cell. But look at these large, dark blue to black granules. You almost can't even see the nucleus. This is our basophil. And when we leave the circulation, then we call that a mast cell. Here we have another peripheral blood smear, and with these you can see how the erythrocytes are stacking something like coins. They come in stacks. This is called the rouleau formation, and we typically see these as red blood cells are squeezing through a capillary or in a tightly packed area like we see here. In this particular field, we can fairly easily pick out our neutrophil. We can see the small granules. We can see the larger, more pink-colored eosinophil granules. But then we have these other cells that are mostly nuclear material with a very small rim of cytoplasm, and these are much smaller than we see with our leukocytes. These are our lymphocytes in peripheral blood. Now we're shifting gears, and we're going to look at bone marrow smears. And these bone marrow smears are going to look at the different developmental stages of red blood cell and white blood cell development. So as we look at this particular image, it's pretty easy to pick out what we can recognize as a neutrophil. Again, we can see the multiple lobes of the nucleus. There's a bar body just sticking right off the end of that nuclear material. So that is our polymorphonuclear neutrophil, even though we are in a peripheral blood smear. These are some very early stages of blood cell development. We're not going to name these particular ones. But then as we look over here on the far left, again, we recognize the granules, small, neutral in color. So this is going to be of the neutrophil lineage. But as we look at this bent chromosome material, this chromatin, we're going to see that it is indented, but it's indented to the point that we really start to have a horseshoe shape. It's difficult to see in this one. So this is going to be one of those gray area where Will you call this a metamyelocyte, which you have that really the indention that's beginning in the nucleus, or do we call this a band or stab cell where it looks more horseshoe shaped? Sometimes it's really difficult to see in the plane of section. This looks like a small indention, so I would have a tendency to refer to this as a neutrophilic metamyelocyte. However, in some of these slides, these authors call them different things, so we'll see what pops up here. In this particular field, we're going to have a fairly round nucleus, so we haven't really entered into the metamyelocyte lineage. The cytoplasm appears to be granular, but it's a darker material, and if you look in between this darker material, you can see some of these pink to red granules. So the specific granules are starting to build up. So this is going to be of the eosinophil lineage, and since we don't see anything in the way of indentation, then we're going to call this an eosinophilic myelocyte, the first one where we can recognize specific granules. Now, as we look at these other stages, these are going to be stages of red blood cell development. And we can see that in this one in particular, do you see how we're starting to get more of a pale pinkish color? Now we see what our erythrocytes, the color of that cytoplasm. And we can have fairly bluish cytoplasm because remember, as erythrocytes go through their process, the cell itself gets smaller, the nuclear material gets smaller and more condensed, and we begin to accumulate material, hemoglobin, in our cytoplasm to change color. So the majority of what we are looking at in this field, because of their size, because of their condensed state of the chromatin especially, most of these are going to be polychromatophilic erythroblasts. Certainly this is a poly. We can see that mottled texture there. These are polychromatophilic. This one is going to be on that boundary between what I would call poly and ortho. You can still see a mottled look to that, but we do begin to see some of the pink, just very beginning stages of this turning pink. So it's possible this is going to be considered an orthochromatophilic. Now again, on your practicals, this is one of those gray areas, much like we see here, that I'm going to do my best to avoid. So in this gray area, they do in fact call this one an orthochromatophilic. We'll see better examples where this nucleus is just jet black. I like that for orthochromatophilic. And over here they do call this a band cell, but in this plane of section, that is difficult to see that we've got a horseshoe instead of an indentation. And then we can see our polychromatophilics scattered throughout. Here we have another field, and what we're going to look at in this is also different stages of our blood cell development. So again, we can fairly easily recognize our neutrophils. Certainly we have our bar body sticking out here, so small granules, multiple lobes, that is our polymorphonuclear neutrophil. But then as we look at some of these other stages of development, here we have the beginnings of a leukocyte. Notice how dark blue these are. These are going to be typical of our red blood cell developmental stages. Now we're getting this lighter color, small granules. Here we begin to see possibly an indentation, but not really. So this is going to be likely a neutrophilic myelocyte, the first stage that we're really going to identify from the bone marrow. But if you look in this particular example, you can see that we've got this jet black nucleus and actually is being extruded from the cell. That's the color I like to identify if I'm looking for a orthochromatophilic erythroblast. But this one being extruded, that's a late stage just before we get to a reticulocyte. Now, most of everything else we see in this field, certainly as we're getting smaller in size, we can still see the mottled nuclear material in each one of these. These are going to be our polychromatophilic. And you can see this is starting to get a little pinkish and that nuclear material is really getting dense. But again, for me, I would still call this a polychromatophilic. You know, they didn't go with what I liked on the other one, so we'll see what they say here. All right, so they in fact do call this one a polychromatophilic along with all of these others, and they have them numbered from the earliest stages to the later stages. So this is an early polychromatophilic all the way down to some of our later stages, 7 and 8. I would have thought that would have been the latest one because of how pink that cytoplasm looked. Again, earlier stages of red blood cell development, the basophilic erythroblast, that dark blue material, proerythroblast, we're not going to be identifying that in lab. That is the first of our stages of red blood cell development. All right, so now as we move along through our list, here again we can see stages of white cell development. And most of these are going to be of the neutrophilic type. We can see these very fine particulates here. So this is going to be of the neutrophil lineage, and that's really the easiest thing to pick out. But here we can easily see these larger pink granules starting to have more of a bilobe nucleus, and you can see just that slight hint of connection of chromatin material. So this looks more like our mature eosinophil, polymorphonuclear eosinophil, than anything else. And then here you see the jet black. You see the same colored cytoplasm as we have with our erythrocytes. That's going to be our orthochromatophilic erythroblast. That's what I like to identify. You see there's no modeling to the chromatin there whatsoever. That's just jet black. Now, as we look at these different ones, we can certainly see an indented nucleus here, an indented nucleus here. So these I am leaning toward calling, we know they're neutrophilic, I'm leaning towards calling these a metamyelocyte. Here I think we have just a full-blown neutrophil. You can see the multiple lobes of that one. But here we're really starting to get a horseshoe shape. So again, this is one that some people might call a metamyelocyte. I might even lean toward calling this a band or a stab cell. So these are going to be some of those stages, that earlier metamyelocyte, either a late metamyelocyte or an early band cell of the neutrophil lineage. All right, so this particular author does in fact link all three of these together, but you can see how distinctly different that indentation is. Hardly any indentation here. You can see a nice division there, and I certainly would agree that these are meta. But that one, that's going to be one of those gray zones that I'm going to try to avoid for your practicals and quizzes. Here we have varying stages of development. And so what we're going to be looking at here, a very early stage development. This is going to be a promyelocyte. This is not one that we're going to identify. But notice this is not as bluish as we have seen for our erythrocyte lineage. This is going to be one of the early stages of white cell development. What do you think this is based on what we have seen and we've been working with? One of the easiest ones to identify. Multiple lobes, light small granules. That's going to be our polymorphonuclear neutrophil. And then over here, and that's tough. You can barely see any modeling. But since you do see some modeling here, this is either going to be a very late stage polychromatophilic or an early stage orthochromatophilic erythroblast. It's fairly small as well. Now here we definitely have polychromatophilic erythroblast. You can see sort of that bluish cytoplasm. We're starting to turn more pink in color here, and you can see this nuclear material is not nearly as dense as we see here. But this is not jet black either. All right, so we got our neutrophil, we got our polychromatophilics, and they didn't label that one. So I like where we were, late polychromatophilic or early orthochromatophilic erythroblast. In this particular field, we see several stages of neutrophil development. Certainly we can pick out our mature polymorphonuclear neutrophil with its multiple lobes, light staining nuclei. And actually we can somewhat go backwards as we look at this. This is our mature neutrophil. Then we're going to go backwards to our band cell, and we can see the extreme indentation here, neutrophilic granules. And then an earlier stage there would be our metamyelocyte. So this is a neutrophilic metamyelocyte. And then beyond that, we can just go to our neutrophilic myelocyte. You can still see some of these very large dark granules. This is a promyelocyte, by the way, and you can see all the dark granules almost obliterating this nuclear material. These are all the azurophilic granules. Well, we're starting to lose those in favor of those smaller fine-grained particulates, and so that's going to make this a neutrophilic neutrophilic myelocyte. Here we also have a mature polymorphonuclear neutrophil. And in this case, we can see the indented nucleus, but we see still somewhat of the granular nature to these and maybe some larger pink granules. So this is going to be a metamyelocyte, not neutrophilic. You see how fine-grained these are. These don't appear as large as the basophilic, so just guessing in the middle, I'm going to have to put that in the eosinophilic lineage, so an eosinophilic metamyelocyte. Then we also have this extremely dark, solid black ball, fairly reddish cytoplasm, and what might you call this? The size, the dense color, that's an orthochromatophilic erythroblast. So I think we got all of our stages correct. We can see our polymorphonuclear neutrophils. We've got our neutrophilic band, neutrophilic metamyelocyte. We have our neutrophilic myelocyte, our promyelocyte. Oh, they didn't label this one. I'm still going to call this one an eosinophilic metamyelocyte because of the indentation. Now, this one's pretty easy. We can pick this one out. Look at the large blue granules here. That's going to be indicative of our basophilic lineage. Fairly large. We still have a round nucleus. So this is going to be a basophilic myelocyte. And that's why on that previous slide, I didn't really want to call this one a basophilic, because you can very easily recognize those basophilic granules that we have. Certainly of the erythroblast lineage, still some modeling, but how small that is and how small this one is. Probably going to call these maybe early orthochromatophilic here. Certainly polychromatophilic erythroblasts there. But they only label the one. Now, in these bone marrow smears, you can often have a large number of cells in a particular slide. And so as we look at some of these images, we have to look for some very specific and characteristic features that we're used to seeing. Certainly in many of these, we can very easily pick out that very light staining cytoplasm. So as we look at this light staining cytoplasm, and we've got this indentation that we have into the nucleus, these are going to be our neutrophilic metamyelocytes. Down here we have this really elongated chromatin material, fairly strong indentation. This is either going to be a late metamyelocyte or maybe an early band cell. And again, you can see how these can get somewhat tricky to pick these out. Really dark staining nuclei here are orthochromatophilic erythroblast, polychromatophilic erythroblast. Certainly you can see the distinction between these two. But now as we look at this large cell, do you see the pink or kind of reddish stained granules in here? Quite a few of them in this place. This is going to be of the eosinophilic lineage and no indentation of the nucleus. That's going to be an eosinophilic myelocyte. We can also see the promyelocytes there, and you can distinguish those from our specific myelocytes. All right, once again, a number of cells. The easiest one to pick out for us, orthochromatophilic erythroblast, certainly the easiest. Here we have a neutrophilic lineage cell, and the really drawn out material. This one's close to being a mature neutrophil, but we may go ahead and just call this a neutrophilic band cell. Here we have an indentation in our nuclear material. That's going to be certainly our metamyelocyte from the neutrophil lineage. Multiple lobes to this one. That's just going to be our run-of-the-mill mature neutrophil that we find in peripheral blood. But now we get here, and again, we can see those large pinkish reddish granules. Again, when you talk about pink and red, compare that to the erythrocyte color. This almost looks orange, but that's the same color that you see here in a particular stain. No indentation to the nucleus, so an eosinophilic myelocyte. Now here, again, this is getting hard because we're losing a lot of those really coarse azurophilic granules. We haven't gotten to the point that we're really talking about a neutrophil yet, but we're starting to get some fine graining in here. So I'm leaning towards calling these neutrophilic myelocytes. There is a slight indentation there, but it's either a very late neutrophilic myelocyte or an early metamyelocyte for that particular stage. And again, those are the stages that I'm gonna avoid for the quizzes. Okay, there in the center, they do call both of these myelocytes, but you can see how different they look. You know, eccentric nucleus, slight indentation. So there are some black and white areas, but there's some gray areas as we move through these developmental stages. Now, as we're coming to the end of our bone marrow smear blood study, we're going to look in the marrow, and we're going to look for some specific examples of some cells that are probably the easiest to identify. This is what it would look like in a bone marrow sample. The smears that we have, of course, we're taking these cells out of the marrow, putting them on a slide, spreading them out so they're easier to see. This is just a section through marrow, and we have this extremely large multinucleated cell in the middle. Let's take a little bit higher mag at that inset. Here you can see this cell, just huge compared to all of the other developmental stages of cells here. Multiple nuclei. Now, there are other multinucleated cells in bone marrow, but remember, they're going to be associated with the bone itself. Those are the osteoclasts. These are even bigger than osteoclasts, and these are going to be the megakaryocytes. Looking at another example, we'll pull this inset up, look even more closely. Here we can see different stages of our red and white cell development, and then over on the right, extremely large multinucleated megakaryocyte.